Thanks very much for giving the opportunity to speak here. So my name is David Gilham. I'm the Chief Scientific Officer at Celyad Oncology. My role is really to help deliver the scientific strategy that reads through into our clinical activities, which is very much focused on colorectal T-cell therapy. We're very much in the Immuno-Oncology space. We've been working since 2016 in this area, and really focusing across a wide range of indications using one specific target, which is called GT2D, a natural killer receptor we're using in the T-cell format. But we're also now developing platforms that allow us to generate really the next generation of CAR-T-cell products beyond our NKG2D receptor. So NKG2D is known as natural killer group 2D protein. It's a protein that NK cells use to target a broad range of stress cells. They do this by targeting 8 different ligands. Now, NK cells are being explored as an Immuno-Oncology asset themselves, but it's very clear that NK cells do have some challenges in terms of the therapeutic context. They have a range of inhibitory receptors that prevents them from activating efficiently just against the NKG2D targets. And they also have some other technical issues, such as real issues around cryopreservation. So the concept is to use this receptor with its broad specificity, but use this in a T-cell. And specifically for the talk I gave during the CAR-T-cell conference, this is in an allogeneic CAR-T-cell. So we're looking to endow T-cells with the potential broad specificity of an NK receptor and allow us to really target a broad range of tumors. Now in the talk of the CAR-TCR, this product, which we call CYAD-101, is a non-gene edited allogeneic CAR-T using the NKG2D receptor. It's non-gene edited. We co-express a peptide with the CAR, which generates the allogeneic CAR-T cells. We think this gives us a major advantage in terms of simplicity of generating these allogeneic cells. And importantly, we've now treated 15 patients. We've seen evidence of clinical responses, some partial responses and stable disease with a disease control rate of around 70%. And as far as we can tell, at least at this early stage, an improvement in terms of the progression-free survival of these patients. Now, importantly, these patients in this trial were metastatic colorectal cancer patients, and they're all microsatellite-stable. So microsatellite unstable patients have been shown to be responsive to checkpoint inhibitors, but the microsatellite stable population are not responsive to the checkpoint and represent a very large population, around 95% of metastatic colorectal cancer patients who have passed 3rd or 4th line therapy. So we think this is very high unmet need. And using this new allogeneic non-gene edited approach, we think gives us an opportunity to really look to try and provide some options to these patients with no other therapeutic opportunities.